Hello, my name is Benjamin Pepper. I'm a sophomore chemical engineering student in Professor Shelley Payton's lab at the University of Massachusetts Amherst. The project that I will be discussing today is all about measuring astrocyte response to mild traumatic brain injury, or mild TBI. So, traumatic brain injury can happen in three different ways. The most commonly thought of concussion, in which someone hits their head hard against something, is known as direct impact injury, with all impact being concentrated in one area. When someone is involved in a car accident and experiences whiplash, they experience the second type of TBI, an acceleration deceleration injury. As their head whips forward, the brain hits the front of the skull, sending it backwards to collide with the back of the skull. This causes the impact to be felt in both the front and back of the brain. In our last type of TBI, blast injury, a shock wave, such as from a bomb, goes through the brain and leaves lots of cavitation bubbles. Cavitations are also seen in the other types of TBIs, illustrated here in these red areas on the figure. They form when there are regions of high pressure differences in the brain, and when this pressure gets to be too high, the bubbles burst and they cause brain damage. Now, after cavitation occurs in the brain, astrocytes, such as those pictured here, transition from an unreactive quiescent state to a bushy reactive state. As you can see, they go through morphological changes in this and secrete and express different proteins as a way to act as a sort of scar tissue and help really repair the brain. So we all know about how tau protein accumulation in TBI leads to brain damage. But what about the astrocyte proteins? The research question I really wanted to look into was how can we visualize this astrocyte morphological change and monitor protein levels in response to mild TBI? Can we prove that these changes occur due to brain injury and are linked to neurodegenerative diseases like Alzheimer's disease and Parkinson's disease? So, to model cavitation injuries seen in TBI, needle-induced cavitation, or NIC, was used. It works through blowing a bubble into material using a needle that will grow and grow until it collapses. This sends a microscopic shock wave through the material as a great way to induce a cavitation injury. This setup shows what our NIC setup looked like in practice, and this is what the material looks like after the cavitation event occurred. As you can see through these, I used collagen hydrogels instead of real brains. So I really did this so that we could see the astrocyte morphological changes that come with the cavitation injuries. As you can see in these images, collagen hydrogels are relatively clear and give us a really just good view that isn't cluttered by other cells of exactly what happens to the astrocytes in cavitation. My goal in this research was to optimize the collagen gel size for use in NIC and to visualize how the cavitation process affects the astrocytes. I did this by using 500 microliter 4 milligram per milliliter collagen gels, as they were large enough to induce the injury, but small enough as to not waste too many cells. The first mold I worked with was an optimized mold that had a 19 to 15 width to height ratio, being 9.5 millimeters wide and 7.5 millimeters tall. Its Fusion 360 schematic can be seen here along with what the mold looks like with no gel. In this picture, the gel has been left out of the PBS for an extended period of time and it appears to have flattened out a bit. So due to this, I made the next mold I worked with a taller mold with a 4 to 5 width to height ratio at 8 millimeters wide and 10 millimeters tall. Its Fusion 360 schematic can be seen here, along with what it looks like with no gel in it. The gel from the mold seen here has been left in PBS and the increased swelling can really be seen. So, from literature, we know that the Young's elastic modulus of the brain is 1 kPa. For gels, it will be slightly different since it doesn't really have all the various cells and the products from all the cellular processes being produced in it that the brain does. However, in a paper by Raub et al., it can be seen that gels follow a general trend. In the graph of this trend that you can see here, for acellular cross-linked 4 milligram per milliliter collagen gel, we are looking at an elastic modulus of about 1.8 kPa. Now, when we do NIC, we get critical pressure readings at a maximum pressure, not modulus readings. So the next big question was how to convert Young's elastic modulus to critical pressure. The equation seen here was used, and where P sub C is the critical pressure, E is the Young's elastic modulus, R is the radius of the needle, and gamma is the surface tension between the fluid and the material, in our case between collagen gel and water. However, collagen gel is mostly water, so the term can be neglected since they are pretty much the same. This simplifies the equation E equals 6/5 P sub C. Now looking at this early tall mold NIC I did, you can clearly see where the cavitation started right here. And the pressure steadily increases up and up and up until this critical point pressure of 3.611 kPa. This value is too high and I'm working on getting it down. But you can see that once this critical pressure is reached, the bubble completely collapses, the pressure drops, and it levels out. So these are the results for my recent optimized mold and tall mold NICs I have done. They don't look as good as there's no distinct cavitation event as before, as you can see through these flat lines and not this like right down as we saw before. But I'm working on replicating the earlier results I saw. So from our work, it can be concluded from this data that the Young's elastic modulus should be about 1.8 kPa for 4 milligram per milliliter collagen gel, which corresponds to a critical pressure of about 1.5 kPa. We can see that the shape of the gel definitely changes its properties, but we need to do more testing to confirm exactly what dimensions best model the brain. Looking to the future, I'm working to load the astrocytes into my gels and see how their morphology really changes due to NIC and quantify protein levels from this. And these images that you can see here, astrocytes have been grown in brain gels. This is very promising news that shows a proof of concept that what I'm trying to do can truly be done. There's a really bright future for this project. So I would like to take a minute to sincerely thank the Peyton Lab Group for allowing me to do research in their lab, especially Professor Shelley Payton and Carrie Dugan. Thank you all very much for listening to my presentation.